Today I'm going to present the g.PANGOLIN system, and Pauline and Matteo are also going to show her work they did at g.tec for the master thesis. I'm going to give you a little introduction about the pangolin system in general. Afterwards, we'll show you some research outcomes, and in the end, while Matteo and Pauline are presenting, I'm going to prepare a live experiment, and we'll run shortly a live experiment. Maybe you've seen before the setup that we did, 512 channels visual setup with the pangolin system. So in general about the system, the g.PANGOLIN system is an ultra high density EEG system with flexible PCBs. So these electrode grids that are attached with medical adhesives to the scalp of the shaved subject can also be used as a EMG or ECG biosignal analysis system, and we use preamplifiers for a better signal to noise ratio, and we can use 256 channels per high amp and also merge several high amps. This is in general the system overview, so we can use up to 1024 channels if we merge 4G high amps. And the inter-electrode distance of the PCBs of the electrodes is 8.6 millimeters, and we have 5.9 millimeter exposed sensor diameter that we fill up here these adhesives with conductive gel. So if you compare the layout of the pangolin system with standard systems, you get something like that. So if we cover the scalp with 64 pangolin grids, we get this setup compared to the 1010 or the extended 1010 system we have here. Of course, a very much improvement of the spatial density of sensors. And generally, the process how we use the system is that we have to prepare a montage, a layout to also get the real positions of the electrode grids compared to the standard positions. Then we apply the grids and the preamplifiers, and we run our experiments. It is also for our experiments that we did important that we got co-registered the positions of the electrode grids, and we got the MRI of the people using the system, so we can also perform source reconstruction techniques. Here you can see that the reconstructed brain model that we used also for one subject, and we used these MRI markers to locate the standard position, so we knew where to place the electrode grids with our grid placement tool, so we can then find the real positions of the sensor locations. And why do we actually need more EEG channels, and why more density? So there is research going on that, especially in source imaging from epileptic patients, it is a very high improvement with higher density of EEG channels. Also, other studies show that, and especially when it comes to spatiotemporal aspects of visual processing, there are also studies investigating that. So that's a good base. And we then concluded a lot of research already with the system, and it's ongoing. And one of the first I want to show you is that increased spatial resolution reveals separated EEG activation of individual finger movements. This was a finger tapping experiment where each individual finger was tapped, so motor execution. And we compared, in this case, with a standard 64-channel EEG cap, our ultra high density EEG. And we used ERDS as a feature, and we checked the statistics and plotted the significant channels that we found for the different finger movements. So you can see here comparison of the ultra high density and the region of interest in red. And on the right side, you can see a standard EEG setup, just the red marked electrode positions, which covers in both cases similar area. But on the right side, you see with 9 channels, and on the left side, you see 73 channels with the ultra high density EEG. So with that, we then performed these individual finger movements, and we found the significant channels for each finger. So you can see them here on the left side, plot on the standard locations that I showed before. And also with this 3D head model, we plotted the significance and with the diameter of these bubbles, the z-score of these values. So we have here thumb, index, middle ring, and little finger, and one channel that concluded all of the finger movements. And on the right side, you can see the results for the ultra high density for 2 subjects, S1 and S2, and the EEG. So we see here the plot of the significant channels, which shows for the ultra high density a focal spot, and the region of interest is 73 channels. Therefrom, we have 14% significant. Compared to the EEG subject, we have 89% significant, which means we have here a very broad area of activated channels. So we can also see here that this spatial resolution improvement shows us this focal spot with the significant channels. And further on, because that's often very important also, people ask what is the correlation of the electrodes, and we compared it here with the 1010 extended 1010 ultra density. You can see the sensor distance 8.6 for the ultra density and 35.4 millimeters for the 1010, for example. And we see here the Pearson's correlation, so the R squared value here, and we can see even though the sensor distance increases very much, like here 4 times, the correlation just increases 2.5 times. So we see here definitely improved content in information in the sensors that we have. So with that, we also concluded other experiments, for example, here the hand gesture decoding, where we did hand gestures like rock, paper, scissors movements of the hand. We did both motor execution and motor imagery. And then we plot topographies and did a classification. So here you can see the coverage, just shortly, central and also on one hemisphere, on the contralateral, of course. And you can see here the topography plots of this investigation with the timing and the task onset of all the 3 gestures, and for subject S1 and S2. And the accuracy in this case increased to around 78.9% after 1 second of the movement onset. Here I have to say subject S1 did the motor execution and subject S2 did the motor imagery, even though we had here significant results for subject S2. But the best was here for subject S1 for the classification pair rock versus scissors. And the system can also be used, of course, to feed source reconstruction algorithms. So it's very interesting, a higher density. It's in literature, it can be found that there is a need for high density for source reconstruction. And we implemented here the pangolin with the field trip toolbox. And we used somatosensory evoked potential from a median nerve stimulation. So here the goal is to solve the inverse problem. And we used, in this case, 256 channels. But of course, I have to mention that in further investigations, the whole scalp would need to be covered for best results of source reconstruction. In this figure, you can see how it looked like. So the setup in the left figure A shows how we did the median nerve stimulation and recorded centrally these 256 channels. And in D, you can see here this phase inversion of when doing the median nerve stimulation from the sensory to the motor area, post and pre-central to this ventral sulcus. And we use that time-locked SEP information and the electrode positions and the MRI to build the volume conductor model. And with that, we then created the inverse solution. And here you can see with the dipole fit method, we found here for right hand median nerve stimulation, the position here in the sensory cortex on the left hemisphere for this subject, which is yeah, the expected area to it to happen, but there are other methods also that can be applied. And as I said, there should be a bigger coverage of the scalp. And lastly, the two biggest projects we did, so we measured again with this individual finger movement, 5 subjects, and you can see here also the topographies and with the timing, as you've seen before, and with the accuracy. And for the index versus ring pair, we get here 79.2% accuracy for this classification pair. And in general, you can also see here on this, the right top, the focal plot of the significant channels, where you can see the time frequency map of thumb, index, middle ring, and little finger. And then as an average, we found for this pairwise classification 70.6 accuracy, 70.6% accuracy for in this study for individual finger and decoding. This was motor execution, and we did 5 subjects in this case. And second study is, as you have seen before, with this median nerve stimulation. We have done here mapping of the central sulcus using the ultra high density EEG system. With this non-invasive system, we have shown that we can get an accuracy of 95.2% locating the channels post and pre-central, the central sulcus, by using here these somatosensory evoked potentials that you can see in the left bottom. So you can see here this head model with 10 channels plotted across the motor and sensory area, and the central sulcus located in between. And you can see here now how this phase inverts of the EPs. So with that, we can also perform different algorithms like spectral clustering and peak detection, as you can see here in the right bottom. And with that, we could classify here sensory or motor, as I said, with a ground average of 95.2%. And on the right top, you can see how the setup looked. So again, centrally located for all of the 5 subjects. And we used here again the MRI to find the exact location of our grids, which is obviously very important to here see the exact sensory and motor area. So with that, with a short intro, I can hand over to Pauline, and I will switch on my camera and work here with Patrick on this visual experiment. And when Pauline and Matteo did their presentation and they give me the go, I will start my experiment here and do a live stream of the measurement. I will just now switch on the camera. Pauline, are you ready?
Yes.
Perfect. Then you can share your screen. I'll stop my share and you can see it.
Yeah, so hi, I'm Pauline, and for my master thesis, I researched different kinds of grasping movements using the pangolin system. The original paradigm was by Andreas Perlea and colleagues, where they researched different types of grasping movements on different kinds of objects and how they were represented in the brain, so that later on this information can be used for BCIs or even neuroprosthetics. And here you can see the different type of movements we included. So we have a power grasp, precision grasp, and pinch. We used a sphere-shaped object and cylinder-shaped object. And for both objects, we had a smaller version and a bigger version, so 4 objects in total. The objects were shuffled between the blocks and in random order. And within the blocks, the grasping movements were also randomly presented. Here you can see how the trials look like. So we had a 2-second preparation phase, followed by the instruction. which was presented on the screen, and then the participant starts executing the movement on the given object holds it there until the relax cue is given at 4 seconds. And every grasp and object combination was executed 54 times, spread out over multiple blocks. And to gain a better understanding of these movements, it's very interesting to study this with higher spatial resolution on both EEG and EMG. And we recorded on average with 236 electrodes EEG placed mainly on the motor areas, and we had 96 EMG electrodes spread over the extrinsic muscles on the arm and the intrinsic muscles on the hands. So that gives us in total, on average, 332 electrodes per participant. And in addition to that, we also had a lead motion camera to study the movements of the motions, and we also recorded maximum voluntary contraction to normalize EMG signals later on. And here you can also see a nice illustration of the paradigm we used when it's executed with the pangolin grips also. So here we see a nice example of how the grasping movements is represented in the EEG over time. So at 0 seconds, we see the cue is given to perform the movement, and then when the participant starts executing the movement, we see nice ERDs coming at C3 and C4 areas. And then when the grasp is held, the ERDs fade again, but for the release, we see another ERD to go back to the resting position. These topographies were made for 13 to 30 frequency. And later on, we have some more plots to show differences between classes. We recorded with a lot of different modalities. So this graph nicely shows what kind of information that gives us over time. Here at the bottom, we have the timeline. So from a -1 to 8 seconds, and at 0 seconds, the trial starts. At 4 seconds, the trial ends. Here again, we have the ERDs. And then below that, we have the EMG signals, which gives us more information about what's happening in the muscles. We see a bit of reaction time here. So around 0.5 seconds, the actual movement starts, and we first see it in the extrinsic muscles on the arm, which are involved in lifting of the arm. And then we have the intrinsic muscles to actually that are involved into the finger placement. And then participant holds the grasp there until the end of the trial when the release happens again. So one row below, we have some information from the motion camera. There's a lot of information you can extract, but this particular channel gives us information about the vertical movement of the hand. So here you see, again, a bit of reaction time, but here the hand is lifted towards the object, and here at the end of the trial, it's removed from the object back to the table. And these patterns also here help us explain how our accuracy also behaves over time. So we classified EEG and EMG, and here at the bottom we can see both EEG and EMG classification accuracies over time. In red we have EEG, for which we used a 2-class problem within one object, and you see that at least a 2-class problem, the chance level is around 50%, and it starts also there, and then once the movement starts, the accuracy also increases to around 75%, but when the ERDs fade, the accuracy also drops, and then at the end of the trial we have another accuracy peak for the release, but with our analysis we only looked for within the trial. For EMG we have a 3-class problem, so we classified all 3 movements within one object, and the axes are on the right. So again, you see it starts around chance level, but once the movement starts, as you can see in the other modalities, the accuracy increases to around 95%. And in contrast to EEG, it stays there until the end of the trial, and then again, the accuracy drops. So on the right, we see some more topographies, and you see the different classes, and we took the ERDs for when they were most visible, so around 1 or 1.5 seconds. And we saw for some participants, the power grasp really had an intense ERD, but for other participants, it was more consistent. And here you can also nicely see some differences between the classes in location or spread. Then if we go to EEG accuracies, for the classification here, we used A to 30 Hertz using LDA, and we classified within objects as a two-class problem. For one participant level, we had around the best accuracies around 69%, and for the objects, this was similar. If we look at the accuracy graphs then over time, we see that the accuracies peak around 1 and 2 seconds, so when the ERDs are most visible. And for this class particularly, it was around 75, 78%. And all these accuracies mentioned here were the best accuracies within the trial. And if we then look at the topographies at the same time points, we can also see that we can nicely discriminate between those movements. For the EMG classification, the accuracies were, of course, higher. And we looked at the three-class problem, so we combined all grasps within one object. And on average, the accuracies were above 90%. And we had here for cylinder, you see again that the accuracy over time, it has a bit of a delay because of the reaction time. But then once the movement starts, the accuracy increases to around 95% in this case. And here on the bottom, we can see the combined EMG signals over time. And you see it for the different grasping movements. So to summarize, we studied different types of grasping movements using the pangolin system. And we saw that even with not super complicated classification methods, the results already look quite promising due to the high resolution we used, as we on average had more than 300 channels. And I'm not finished with the analysis yet, so I hope to even improve the results further so that we can hopefully contribute to the research in grasping BCIs or even prosthetics. So I think then if Matteo is also ready, I can stop sharing.
Pauline, thank you. Thank you very much.
Okay.
Nice to meet you to everyone. I'm Matteo, and I worked on my master thesis with Pauline and Leo on g.PANGOLIN. So in this case, I will talk about the benefits of covering a wider region of the scalp while we use g.PANGOLIN. The idea and the concept is to find some other places on the scalp which are relevant while we see a motor execution task, or for example, in this case, also a visual task. I will start by talking about motor execution and motor imagery task for my subjects. I had 4 subjects, and right now I'm still working on my thesis, so my results are not completed. But the idea of the paradigm was having blocks of runs composed by 4D trials, 20 per class, and they were randomized on their order. So the subject didn't know which one was the next one. The idea was to lift the thumb finger and the pinky finger. So the idea was to discriminate the differences between the lifting of these 2 fingers. In front of the subject, there was always one avatar that was emulating the real task. So the idea was to do an online classification to get the results and then a prediction from the avatar. So the idea was to have different coverages. I started from the regular one for one subject, for example, and we can see that the difference between all the coverages is about the spread of the montage. For the first one, for the regular one, I covered also the temporal region. Then on the reduced ones, like the reduced montage on the super reduced, I just focused on the sensory motor region, especially for the super reduced montage. So the idea and the focus on the thesis was to check if there was any other activation on the temporal region, for example. And this is the comparison between the offline classification of my results for one subject, for example. The idea is that here you can see that there are the results for every coverage. The peak is always quite similar on the spread of the coverage. The difference is about the second peak that we can find during the maintaining of the movement, so around 3 seconds for motor execution, and also for motor imagery, but a little earlier, around 2 seconds. So when we see these results, we can see there is a slight difference between covering more on the scalp or less. But it's also interesting to see topographies about it, because we don't have just an idea about accuracy and classification, but also about the topography on the scalp. I need to say that in this case, for this exact moment, so for 1.3 seconds, that it's the exact time of the maximum accuracy. The topography was made by basing all the map on CAR, so common average reference for every montage. So CAR is always different in these 3 montages. And we can see that there are some relevant differences between thumb and pinky, of course, especially on the temporal region for the very first montage, and also on the central part of the sensory motor cortex. But if we go with some less spreaded coverages, we can see, of course, that in the reduced ones, there is less coveraging, so the CAR is less spreaded, and we can see a very nice plot here, the red plot of the activation. But we don't have some nice results about the central deactivation that we had. For the super reduced ones, when I compared just the grids on the sensory motor cortex, we can see that we don't have that much. Also, these results were, of course, related to different scales, because having less channels and using CAR, of course, is quite similar to all the channels. So we can see there is less activation, of course, because of it. So having, of course, a more spreaded coverage helps also for topography. Then I pass to the visual tasks. In this case, is a visual feature task, and I was comparing the stimulus based on colors. We can see we have some bands colored in different ways, from red to green, blue, and black and white. And we can see in topographies for a very complete montage, the visual region, the occipital region that was focusing on the task. So there is a really good definition of the region. And here we can see also the EPs based on the grids that they are colored in different ways. So we can see that always around 0.2 seconds, we have the most activation, so a peak 200. In this case, the classification was made for 3 subjects, and we can see that we always have a peak of our classification around 0.1 and 0.2 seconds, and then a high peak for subject 1 at the end for a P300. It's interesting also to see with confusion matrix the difference in prediction between classes. Seems that always black and white is the most well predicted, and then there are the other colors. My idea was always about it to check, even in this case, what are the differences in covering more with the grids. And we can see for accuracy, in the covering there isn't so much, of course. Sorry, I will show first this, it's more interesting. So let's see first the coveraging. In topography here, we can see very easily the differences. Covering more, of course, makes the region of activation more visible. So we can really see the edges of the activation region. If we pass to a less covered region, like the reduced ones or the super reduced ones, yes, we see some activation, but it's not that good. So here we can see a very nice difference when we talk about topographies. Passing to the accuracy, there isn't so much difference, but I believe that if we want to understand also the source and the region that is activating, is really important to have an idea of what we are seeing. And these are the references for our work. And as I was saying, that the results are still coming because of the working on the thesis. Hopefully in a month, everything will be ready. I don't know if there is any question about my work or the other works. So I can pass.
Thank you, Matteo. I guess we keep questions for afterwards.
Okay.
So I would just now proceed. If you could close the screen share, then I guess my video is shown bigger. So you could also see exactly. So you can also see public right now. So what we are doing are visual experiments. So we have run with different paradigms today. I will show you a bit around how we do the system setup with the Pangolin, and then we'll run an experiment and show you some visual EPs and topographies that we plot online. So I will just go here with the camera to show you the setup that we do for 512 channel visual experiments. So this is here covering the whole occipital region. We perform the experiments in front of the screen, and maybe you can see here. No, you cannot, but I will move the screen a bit so you can. So here on the bottom of the screen, we have mounted a photo sensor, and this one we feed back as a trigger. So we have the exact onset of the visual stimuli, and that's very important to have this hardware trigger for the exact timing of the visual evoked potentials. So maybe shortly to explain again the system setup a bit of the Pangolin. We have here the electrode grid with 16 channels, and here I attached the adhesive already, and the conductive gel is to be here put inside, so we have no shortcuts, and they're insulated. The most important is to prepare the skin, clean it nicely, and then attach the preamplifiers. You can see here the preamplifier cables, and then I will show you a bit around how it is connected actually to the high amp. So we have here these connector boxes. You can see this here. We have one connector box with 16 of those cables. So we have 16 of those cables, means we have in total 256 channels, one high amp, and they're connected here to the high amp. And we use two high amps, so you can see two high amps, two connector boxes, and battery that power our connector boxes. The red cable is in this case the shared ground that we need for both of the amplifiers. And the grounding in general is this cable that we mount here. Second, I'll take this cable so I can show you a bit around. So we mount the ground here on the mastoid with one of those standard electrodes with a clip here on the mastoid. And this is the total setup with 512 channels. So you can see there is still a lot of space left. So we could definitely attach more channels here. But we are interested, obviously, in the visual region, in the occipital region. And if I place the camera like that right now, I can show you nicely how the experiment will look. So I will just go to my computer and start the experiment and show it to you here. So we're running a couple of different experiments. In general, so as you've seen before, these visual features that Matteo showed are different bars with colors and orientations and contrasts. But for this experiment, we just will present faces, so faces of known people in the company, and we'll also present some random objects. And we use this then to plot the EPs, just to show you here the online functioning of the system. I will show it to you first with the camera, and then whenever Christoph can add me with my computer, so the experiment computer, I can also do a screen share of the model. So I will just start now the experiment, and then you should see the screen. On the left bottom where I put my finger now, maybe you can see it, here is the photo sensor. That means we have here a black square, and whenever a picture pops up, we'll talk a bit more quiet, we have a white square, because then the photo sensor reacts and sends the trigger to the system, and we feed back the trigger to the G.high amplifier. And this is important then, of course, for offline analysis as well as online analysis. So we can use this trigger now in real time to plot on an interpolated, so on the head model that we have. Maybe I can show it to you after the experiment finished on the screen, and afterwards maybe if I can share it, that you can see the EPs that we have and also the topologies. Okay, so experiment takes a bit more. And let me check if I can just move myself on the other computer so I can share also the screen with you. This would be important. Maybe if you want, Pauline and Matteo, you can check in the Q&A and already start answering some questions. Meanwhile, I'm trying to share my screen. Would that be okay, Pauline and Matteo? Can you hear me?
Yes,
Yeah,
Leo.
sure.
Sure. Can you go to the Q&A and start answering some of the questions already? This would be great.
Yes.
Yes, sure.
So what do we have? First, please tell the question and then you can answer it and speak it out loud for us. I cannot hear you right now.
Yeah, okay. So I think one of the questions is, what was the highest number of channels that you've ever recorded with g.PANGOLIN system?
Actually, we recorded these 512 channels. Is now the highest amount that we do with 2 high amps in our experiments, but we did like a couple of years ago actually an interesting presentation at the Ars Electronica Festival with the Pangolin scale system, where we covered the whole scalp and we used 4 G.high amps. So we in total covered 1024 channels on the scalp. And yeah, this was quite some effort to set up, but for our experiments, we now try to reduce the setup effort and cover special regions for these locations that we're interested in. So maybe you can do the next one.
Yeah, okay. Another question is, what kind of g.tec product was used to map the topographies of the brain?
Okay, yeah, that's a good one. So it's the g.PANGOLIN montage creator. So there are different 3D montage plots, and we have one for the g.PANGOLIN. So there is this montage gradient, these 3D tools that we can use with the system to plot results and also online. So we have the results in online and then also in our papers. And I will show you also right now how it works. So I'm joining now with my computer that I'm using here, and I will just share the screen in this case. So I hope you can see it. We have here all the EPs from the system, from the experiment that we just recorded. So this visual evoked potentials. If I zoom in on one, I can look for, so in total we have 512 channels. So here, this one you can see a nice peak, and we use this one. So we use these channels, we feed all of the 512 for the experiment. So what I'm going to do now is run it again and show you then online these topography maps that we just talked about. So maybe you can also proceed with the questions, Pauline, and answer them on your side, so I can be quiet and let Patrick focus on the experiment.
Yeah, okay. So one of the other questions is, how do you know you're not classifying muscle artifacts? I think one important step is that you also visually inspect the data, and also using these topographies, you can also see if there's muscle contamination, but also partly you can filter this out by going to certain frequency bands. And I think one question again is how much we'll be providing for a subject in EEG acquisition of motor imagery task. Do you mean that like financial compensation, or what do you? I'm not sure. Or how much time? I'm not sure what this question means, but I think in general the blocks we used for motor imagery were around 7 minutes, and then the motor imagery task was around 2 to 3 seconds. And for some paradigms we used 5 seconds, if I'm remembering it correctly.
If I maybe can jump in here. So what we see is here the topographies of the evoked potentials, and we see here in this area an increased activation. So we go on the EP. Now we probably have some movement of the grounding, and I'll just show you the model as it looks. So what we do is we take these two g.tec and use all of the channels, filter them. Basically, I can also show you here, that's the unfiltered channels. I will show you here the filtered channels when I start the experiment again, and we filter them, select, of course, the bad channels, and then we do a global CAR. It's important, this common average reference, because we record monopolar, and we then use the trigger that I told you. So we take the trigger from the g.tec, this hardware trigger, for plotting these averaged signals with the g.PANGOLIN system. And we use the paradigm presenter. As you've seen, the paradigm is presented with this timing that we set with the paradigm presenter, and we load the pictures there. I will just now make a new scope to show you the data. Exactly, we have here 512 channels, so that's quite something. So I will just reduce it to 50, I would say. And we can go through the channels here. We adapt the scaling a bit. So 100, 100, and then we can see here the channels.
So Leo, maybe I can interrupt here because
Sure.
many people are asking us if there's a difference between neighbor channels if they are so closely together. And if you are now looking at the traces, for example, the top two ones, then you can see the traces are pretty similar, but they're a little bit distinct. This is exactly like EEG data looks like. So also in traditional EEG recordings, we see a slightly different waveform in neighbor channels, and exactly the same is happening here with the pangolin. But the resolution, like mentioned before, is 8.6 millimeters. And this is just super important. Otherwise, we would just collect the same information with all these 1024 electrodes. But you can see just very nicely here that this is not the case. And all the channels have distinct information.
Exactly. So yeah, so these are all the 512 channels that we record with both of the g.HIAMs. And maybe I can show you also, if I load the second paradigm, how these visual features look exactly like that I mentioned before. Maybe meanwhile I'm loading it, you can proceed with answering questions, Christoph.
Sarah is asking, what's the highest number of EEG electrodes you ever measured with the pangolin system?
We had that already before. I guess
Sorry.
the couple of first are already answered.
Okay. And did you also have the first questions already?
Yeah. I guess the first three we already answered.
So I just ask for safety again. Could you also make it dry? Could you work with dry electrodes as well?
Okay, sorry. No, we didn't have that. No, well, basically it's a very complicated topic to work with dry here because we need the exact coverage of this little dot because we don't have a lot of space if you want to place so many electrodes. So for doing that, we actually need here contact with gel on the skin. Being dry would mean that we just place electrodes here on the scalp with no conductive gels, which of course would reduce the signal quality.
And of course, the electrodes must be super flexible because Patrick has a pretty round head, like everybody. They have to attach perfectly to the head. So if the material would be stiff, it wouldn't align well, and then we wouldn't have a solid contact. And this is, of course, super important. So we use adhesives which are very sticky on the skin side to be able to mount it carefully. And the diameter is so small. It's also a reason why the impedance is higher than with standard EEG electrodes. That's the reason why we have the preamplifier here in black, very close to the pangolin grids. Otherwise, you would get a lot of interference and power line noise. And Couton is asking about the price. So for 256 channels, you need a HIAMP, which is around 40,000 euros, depending on the model. And the preamplifiers and pangolin grids and so on, they add on top about 10K. So for 256 channels, it's roughly 50K. And if you want to record from 1024 channels, it's around 200K for the whole setup. But then you get fantastic results.
Now I'm also making you experience the whole experiment that Patrick has to do. So by doing that, as you can see, we have these different spatial frequencies and colors of these bars. And we try to find exactly here that. So we try to classify colors, spatial frequency, contrast, black and white of these bars. And you can see on the left bottom, actually very nice for you, this is the trigger window. So here we have the trigger photo sensor that we then feed back to the g.HIAM. You can see it on your screen exactly there. So just share again and let Patrick do the run. So this is basically also one of the experiments we do with this visual setup. And we could nicely classify these different setups of spatial frequency and orientation of the bars that you could also see. and the different colors that we have. So maybe we can proceed with the questions in this case. So Patrick could run the experiment.
Raji is asking how to get it in India. So on the g.tec AT page, there's a menu item called Find g.tec in your country. So you can just click on the map on India, and then you will find our partner Media Analytica in India, and they can provide you with much more information, quotes, and the prices and so on. So I send you the link as well.
I just saw that somebody asked for the trigger specifically. So maybe when the experiment is over, I mean, I can just point here now. So you can see here this little window that flashes up white whenever there is the presentation. I don't know, I guess you cannot really see this LED on our trick box here, but whenever this sensor is grabbing the white compared to the black, we get a trigger in our trick box. And that trigger with this cable, we feed back to the high-amp, the biosignal amplifier, and with that we have it in our data. So we have the real presentation time of the visual stimulus, and with that we can then plot the APs and also use the APs for classification with a better timing. Because imagine if you just connect the USB cable with the trigger information, you would have a proper delay of the serial transmission if you just save it in the software, this trigger. So the hardware trigger is really important for experiments like this, evoke potentials. So there are any other questions?
Did you already speak about reusing the pangolin grids?
No. So what I can show you actually with the video camera here a bit. We did some experiments. Actually, I did also experiments on me similar to the ones we do with Patrick today. And you can see them here. Clean the electro grids from the adhesive and the gel that we use. And by doing them, they're reusable. So just these PCBs, these flexible PCBs are reusable and can be used in new experiments. We can reuse them a couple of times. Of course, you should clean them careful because they're very slim and thin for that it covers the scalp nicely. So if I go a bit closer, you can see how it's actually attached to the head. We have here this little on this preamplifier, this little foot. So the amplifier is not moving forward and backwards too much, so it's not bending the connector, so it's more stable. We just located here one on C set to have a good orientation, and also on O set, I connected here one. You can see that on location number 1, and with that I actually got my my montage as I did the 3D head model. So according to that, I then just have my plan here with all of the 32 grids that I have, and I attached it according to O set and C set. I aligned them accordingly, and with the geometry of the grids, you can have then the perfect setup and the coverage. So luckily, Patrick in this case has a very nice round head, so the coverage works pretty well. Of course, there are different head shapes, and this also depends how good the electro coverage is and how much distance is there maybe in between grids. Exactly. So that's exactly. We also have here a cable holder for our cables, so they don't pull on the preamplifiers. We just apply them to this holder device. So then the whole setup is more stable, and we can record with this setup a whole day. And of course, we also try to do a lot of experiments in one day because setup time is also, it is, let's say, 1 to 2 hours with preparing everything. Sticking the electrodes doesn't take so much time, but then we try to cover as many experiments as possible. That's what we try to do with the Pangolin system.
Leo Side is asking to show the connector, how it's plugged in into the head stage.
Sure, I can do that. So I will just plug it out now. So plugging it out, you normally do just at the end of the experiment, but you can see, if I film that from the top, you can see how the electrode is attached, and you see this connector with 16 channels. Number 1 needs to be in the front, of course, and then I can just you can see here the connector with the 16 channels. I just need to make sure number 1 is in the front and that it's aligned correctly. And I guess you don't hear the click, but when I press it in, there is a little click. So you're also sure it has the right connection. And that's how we now applied the 16 channels for CSET. So that's how we actually do it with the system. Yeah, and on the, yeah, exactly. Maybe again, because the question was before, with this camera, it's easy to move around. I hope it's not moving too much for you. But you can see here on the back side of the C-HIAM, I'm showing you the trigger cable that I was mentioning before. So you can see here we have power, USB, and trigger. And this gray one is the trigger cable. And it actually goes over here to our trick box. So that's exactly what we need. This trick box with the photo sensor that is applied to the screen here, as you can see. So this is how we get the hardware trigger for the evoked potential experiments. We have our 2 HIAMs connected with USB and USB-C, our HDMI for the screen, also connected here to the computer directly. So with this computer, you can do the experiment.
Leo, there's one question. What can you do if the impedance is too high in one of the channels?
Yeah, so in general, you need to make sure that the setup is properly done so they are attached nicely. You can also work with like a little, maybe I have one here, a pen or one of those little sticks that you can then check the data quality and then also attach channels manually and grids. So when you set up the whole system by applying the grids, you should make sure that all the grid is attached nicely to the skin. So you can tap a bit on it, like maybe like I'm doing now. So I'm just tapping on all of the sensor locations, and then I can make sure they have good contact. But therefore, it's important that you have a channel detection for bad channels, that you can then also remove those from further processing and from your common average reference.
But this little tapping on the electrodes is actually good enough to reduce the impedance, because we have this very sticky electrode gel for the pangolin. And when you just press it a little bit, it gets in solid contact with the skin. And over time, it even gets less because it goes more into the skin. And Leo, is it still possible to show all the EEG channels on the screen? Or are you switched off?
I can run again, yes.
Could we see channel 16 of the pangolin grid, which is over CSET?
Let me try to open the...
I don't know how easy it is to find this channel. Maybe it's the last channel of all recordings. Because beforehand, when you filmed, we could see that channel 16 is a little bit elevated. So it's not perfectly attached to the skin, but there's still this sticky electrode gel filled to the electrode. Now you can just see yourself how the channel looks like.
Yeah, let me just check that again. So I can then touch the channels and you see how it reacts to you. Let me share my screen. here.
But you have just seen how quick it is to assemble 512. You can also see about half of Patrick's scalp is covered at the moment with pangolins. So when I was doing my PhD, I needed about 1 hour for doing a 64 channel assembling. So if you're doing 1024 channels, this would be 8 hours. Now you can see how clean the EEG data actually looks like. So it's beautiful EEG, and only when Leo is touching it, of course, then we get these fluctuations because he's charged up electrostatically, and then we have also the movements of the electrodes. So which electrode is now?
So this should be actually on the back. So I remember the last grid was, but we are doing already a common average over that and with the bad channel excluded. So it's a bit hard to see that exactly now, actually.
So it's 512?
No, that should be number 1. But with the common average reference.
Sure, but the CAR is taking the average of all the electrodes.
Yeah, let me check that.
Maybe you can just show the last grid.
Yeah. It's exactly this one here. So if I show.
You see also this channel is show all the channels. That's very important when you're visualizing EEG data. So visualize about 8 to 10 seconds on the screen, show a couple of channels. They have to look very similar.
There's a couple are not 512, these are 256. I will not show just 16, so you can see exactly one grid.
So this visualization is nice. And when you compare neighbor electrodes, like mentioned before, they should be similar, but not identical. Identical means you get shortcuts. And if they look pretty much the same, then you can be pretty sure that you are recording clean EEG data. And here you can also see that the lifted channel 16 is nicely visible because the sticky electrode trail still has some contact. And the washer is also making sure that we don't get shortcuts between the electrodes, which is of course super important. Then somebody is asking, why are you using a cable to transmit the data and not Wi-Fi or Bluetooth?
Yeah, well, we would like to, but this is a quite high amount of channels, and it's not so easy to transmit 512 channels with a sample rate of 1 kilohertz, for example, with a Bluetooth. But this is also definitely something that is interesting, that these signals are transmitted wirelessly. Also, it's important that you then have a receiver station that can handle the income of so much data, and that is then aligned properly. And so this is quite a hard topic.
It's actually just not possible is the easy answer with 2.4 gigahertz technology. So with 1024 channels, 24-bit, we are transmitting float 32. You need a much, much higher bandwidth. Its technology doesn't exist yet for that. And with this, Leo, are you finished? So
I am, yes.
Then thank you very much for the nice demo. Matteo, Paulina, you are still here. Pauline, sorry.
Yes, of course.
Hey, nice to see you again. Thank you very much for the nice presentation. Paulina, you're also here?
Yes, hello.
Oh, here you are. Thank you very much for the demo and all the explanations, answering the questions. Leo and Patrick, thank you very much. It was a very impressive demo and very nice work you did in the past months. Thank you very much and enjoy the day.